What happens to your sample when it gets sent off to the lab? After the samples are posted by the study volunteers, they are received for analysis. When samples arrive in the lab, we sort them immediately. This is to ensure older samples are processed first, and to ensure the viability of the sample. Every sample that we get at UK Biobank is logged into our laboratory tracking system. This is important so that we can track every sample through the system. Envelopes are then stored at 5 degrees C in our reagent store to better preserve the samples. The samples are then taken into a microbiological safety cabinet so that they can be handled with the least risk to our laboratory staff. While the risk from the volunteer samples is considered low, we take health and safety and the prevention of spreading COVID-19 very seriously. The envelope is then scanned, opened, and the contents carefully removed. The sample tube is then secured into a centrifuge bucket and the survey filed away to be scanned, archived, and reviewed. The centrifuge buckets are then handed over to the processing team. The samples are spun at high speed in a centrifuge for 10 minutes to separate the components of the blood into distinct layers. As you can see, after 10 minutes in the centrifuge, the blood has been split into several layers. The red layer contains heavier parts of blood, such as red and white blood cells. The pale yellow layer is called the plasma. This contains all the proteins found in blood, including antibodies. The serum needs to be removed from the rest of the blood for the test. We use pipettes to carefully draw this layer out. This is then put into a well on this 96 well tray. A second fraction is then taken to store on a second plate as a backup in case further testing is required. A further 79 samples are then placed in wells on the same plate. The first and final columns are left empty so that control samples can be tested. Once the plate is complete, the wells are thoroughly sealed to ensure no loss of sample or cross-contamination during storage and shipping. The identity of the sample in each well is logged into our system so that we know which samples are where. Once the plate is completed, it's ready for storage at -80 degrees C prior to shipping. Each day, up to 1,200 samples will pass through this lab. It's our aim to process them into storage as quickly as possible to maintain the best integrity of every sample. The samples are shipped once a week to a different lab for testing. On behalf of UK Biobank, thank you for providing so many samples for this study. Thank you, and let's stop COVID together.